Good morning everyone. Welcome to my channel Botany with Dr. Mranali Chauhan. Today we shall continue our topic of the last video, loss of water from plants. In this second part we shall discuss about the opening and closing of stomata. As we studied in our last video, the loss of water by the plants in the form of water vapor is known as transpiration. 80 to 90% of transpiration occurs through stomata. Stomata are small pores surrounded by guard cells. When water enters the guard cell, they become turgid and the stomata open. But when water comes out of the guard cells through exosmosis, the guard cells lose their turgor and become flaccid. This leads to stomatal closure. Now, what are the mechanisms for the turgor changes in the guard cells that actually lead to the opening and closing of stomata? Several concepts have been given, and the earlier concepts are known as classical concept, and the most accepted modern concept is the potassium pump and proton secretion theory. So, first of all, we shall discuss the classical concept. The classical concept was proposed by G.W. Scarth in 1932. It has been observed that stomata open in sunlight and in the dark the stomata close. Light initiates the opening of stomata, and therefore this type of opening of stomata is known as photoactive opening. We all know that in the presence of sunlight, photosynthesis takes place, and it was proposed that light mediates through photosynthesis in the opening of stomata. So what happens? As the light falls on the guard cells they contain chlorophyll, and therefore photosynthesis begins. And as the photosynthesis starts in the guard cell, CO2 is consumed in photosynthesis. Therefore, the level of CO2 in the guard cells and its surrounding region, it decreases. CO2 is used in the formation of various types of organic acids in the plant. So as the CO2 is consumed, the concentration of the organic acids in the cell, it comes down, it lowers, and therefore the acids break down to release more CO2 for photosynthesis, and the pH of the guard cells increases. As the pH increases, the enzyme phosphorylase that is responsible for the conversion of starch into sugars, it activates, because the activity of phosphorylase is highly dependent on the pH of the medium. Therefore, as the pH increases, the enzyme it activates and conversion of starch into sugar begins. As the starch is converted into sugar, the potential of the cell decreases, and water from the surrounding subsidiary cells, it enters into the guard cells, and guard cells, they become turgid and stomata open. Thus, according to classical concept, light mediates through photosynthesis. It leads to the consumption of CO2 that ultimately results in the conversion of starch to sugar. The starch sugar conversion, it is responsible for the change in the turgor of the guard cells and opening of the stomata. In the dark, photosynthesis stops. The concentration of CO2 in the guard cells and the surrounding cells, it increases, and this leads to the decrease in the pH. With the decrease in the pH, the conversion of sugars to starch begins, and therefore the water potential of the cell it increases, and water from the guard cell enters the subsidiary cells. Exosmosis takes place and the guard cells they lose their turgor and the stomata close. So the opening and closing of stomata according to the classical concept, it depends ultimately on the interconversion of starch and sugar. As the level of sugar increases, the guard cells they gain their turgor, and as the level of sugar decreases, they lose their turgor. This ultimately leads to the opening and closing of stomata. Several evidences support this opening of the stomata through the starch-sugar interconversion. First, if the pH of the guard cell is increased artificially by addition of agents like ammonia, it leads to the stomatal opening. Second, decreasing CO2 concentration from 300 ppm to 100 ppm induces stomatal opening. 300 ppm is the normal concentration of CO2, but if it is decreased, it leads to the stomatal opening. Vice versa, if CO2 concentration is increased, it leads to stomatal closure. The third evidence is the activity of enzyme phosphorylase. The enzyme phosphorylase it converts starch into sugar, but its activity is affected by the pH. At pH 7, it converts starch to glucose, but at a lower pH, that is when the pH is about 5, then the reaction takes place in the reverse direction, that is sugars or glucose is converted to starch and inorganic phosphate. And the fourth evidence that supports this classical theory is that during wilting stomata closure takes place, and as the stomata close there is rapid accumulation of starch. So from these evidences we can say that the change in the CO2 concentration leads to the change in the pH, and ultimately it affects the activity of phosphorylase enzyme, and all this leads to the opening and closing of the stomata. But there are still some objections against the classical theory. First of all, classical theory is based on the starch-sugar conversion, though there are several plants in which this starch-sugar conversion doesn't take place at all. Secondly, in monocots, guard cells do not form starch. Third, that is specific to tobacco plant. In tobacco, the opening of stomata is actually related to the metabolism of glycolic acid and not that of sugars. And the fourth, that is most important, stomata open more rapidly in light, but the consumption of CO2 in the photosynthesis it takes place at a slower rate, and therefore stomatal opening cannot be accounted for photosynthetic consumption of CO2. Thus, the classical concept that was actually based on the starch-sugar conversion, it cannot be applied to all the plants, and therefore a new concept that is more accepted about the opening and closing of stomata was proposed, and this is known as potassium pump and proton secretion theory. This theory was proposed by the Japanese scientists Imamura and Fujino. According to is concerned, large amount of potassium ion move into the guard cells, leading to stomatal opening. Imamura and Fujino, they observed that as the stomata open, a large amount of potassium ion, they move into the guard cells, and their movement actually leads to the change in the turgor of the guard cells. Several evidences support this potassium pump and proton secretion theory. We shall discuss them. First of all, inward flux of potassium ion in response to light and CO2 free air has been demonstrated in various species. That means in the absence of CO2, inward flux of potassium ion takes place in the response of light, and as the potassium ion influx occurs, the water potential of the guard cell decreases. Therefore, this evidence supports that the potential change in the guard cell is not mediated by CO2. Second, accumulation of potassium ion by guard cells from cell set adjacent to stomata is strongly correlated with the opening of stomata. As the stomata open, it is observed that large amount of potassium ion they accumulate in the guard cell, and this accumulation leads to the change in the potential of the cell. And one experimental proof that was given, and when the leaves of plants they are floated on the solution of potassium chloride, the stomata open in the presence of light. That means if light is provided and potassium influx takes place, the stomata will open. Though light initiates the opening of stomata, but mechanism other than starch sugar conversion take place that is responsible for the opening of stomata. Light mediates the opening of stomata not through starch sugar conversion but through the accumulation of potassium ions in the guard cells. So, according to the modern concept, as the light falls on the stomata, potassium ion influx it takes place in the guard cells. It leads to the increase in the osmotic potential of these cells, and therefore osmosis of water takes place from the subsidiary cell, leading to the turgid state of the cells and the stomata open. But in the dark, potassium efflux takes place, that is potassium ion they come out of the guard cells, and therefore the osmotic potential of these guard cells it decreases, leading to the exosmosis of water, and ultimately the turgor of the guard cell it decreases and the guard cells lose their turgid state, the stomata close. It was seen that the role of potassium ion in the stomatal opening was specific. Other monovalent ions like sodium ion they do not exert the same effect. Means only potassium ion are responsible for the opening and closing of stomata. No other ion can be used in their place. Also, the influx of potassium ion is mediated by ATP. Energy is required in this transfer of potassium ion. These ATP are provided by non-cyclic photophosphorylation by mitochondrial respiration or oxidative phosphorylation. Respiratory inhibitors like azide, cyanide, 2,4-dinitrophenol, they inhibit stomatal opening, leading to the conclusion that ATP generated through respiration are required for the influx of potassium ion. So, how does the procedure takes place? When stomata are closed, respiration takes place and starch is converted to PEP or phosphoenol pyruvate. This PEP makes several types of organic acids with carbon dioxide, and malic acid is the most important of all the organic acids. This malic acid is transported to the vacuoles, and in the cell it releases hydrogen ion that are exported to the subsidiary cells, and as the hydrogen ions are transferred to the subsidiary cells, to maintain the ionic balance, potassium ions are transferred from subsidiary cells to the guard cells. In some plants, along with potassium ion, chloride ion also enter the guard cell. So, as hydrogen ions are transferred from the guard cell to the subsidiary cells, the pH of the guard cells decreases, and thus in this complete procedure, consumption of CO2 and formation of organic acids takes place, and along with the transport of hydrogen ion to the subsidiary cells lowers the pH of guard cells. As the potassium ion enter the guard cell, the water potential of the guard cells decreases, and therefore water enters into the guard cell from the subsidiary cells, and guard cells become turgid and stomata open. So, the role of light can be compiled as following. As light falls on the guard cell, photosynthesis takes place, and this concentration of CO2 decreases because CO2 is used in the photosynthesis. As CO2 concentration decreases, decarboxylation of organic acids takes place, and this leads to the increase in the pH of the guard cells. Phosphorylase enzyme is activated, and the conversion of starch to sugar it takes place. The rise in the sugar level according to classical concern leads to the change in the turgor of the guard cells, but according to the modern concern, this change is insufficient to affect water potential of the cell. Next, photosynthesis in the guard cell, that is mediated to the light. It also leads to the CO2 fixation and ultimately the formation of sugar by photosynthesis. This is again insufficient to affect water potential of the guard cells. Third event that takes place is the production of organic acid, the most important malic acid, and it ultimately leads to the exchange of ions and influx of potassium ion into the guard cells. This process requires ATP, and ATP are provided by photosynthesis through non-cyclic photophosphorylation. These ATP that are formed through non-cyclic phosphorylation, they mediate in the exchange of hydrogen ion and potassium ion. The potassium hydrogen pump is driven by these ATP. ATP are also provided by respiration. So, the ATP formed through respiration and through photophosphorylation, these ATP ultimately lead to the exchange of potassium and hydrogen ions, and as the potassium ion and along with them chloride ion they enter the guard cell, water potential becomes more negative, and this change in the ion concentration of the guard cell, it is sufficient to affect water potential, and so the water enters into the guard cells and osmosis takes place, and turgor pressure of the guard cells increases and the stomata open. Thus we can say that in the presence of light, photosynthesis takes place, starch sugar conversion does occur, but it is insufficient to affect the water potential of the cells, and ultimately the change in the potassium ion concentration, it is ultimately responsible for the opening and closure of the stomata, and the starch sugar conversion that was proposed by the classical theory is insufficient for the opening and closing of stomata. Thus we saw how the light mediates the opening and closing of stomata, why the stomata open in the light and they close in the dark, and in most of the plants stomata opening is photoactive opening, that is initiated by the light, but there are some plants in which the dark initiates the opening of stomata. This opening of the stomata that is initiated by the dark is known as scotoactive opening. The mechanism of scotoactive opening was explained by Nishida in 1963, and most of the succulent plants, they have crassulation acid metabolism, and they are also known as CAM plants. In these plants, scotoactive opening is found. It was seen that during the daytime, the pH of the cell sap in these succulent plant is very low, and in the evening it is highest. This change in the pH of the cell sap is known as diurnal acid cycle. That means that there is a change in the acid concentration of the cell using radioactive carbon 14 containing CO2. It was found that malate is fixed during the night in these plants, and this malate, it leads to the lowering of the pH, and this fixation of carbon dioxide in the form of malates is known as dark fixation of CO2, and the CO2 that is fixed in the form of malate is used during the daytime in the photosynthesis. Actually these plants, the succulent plants, they are xerophytic plants. They are found in environmental conditions where there is high light intensity. Light intensity is very high and availability of water is low. So these plants have a special mechanism of stomatal opening that is an adaptation to conserve the water, as light intensity is very high in the desert areas where these plants are found. So in these plants, the stomata, they don't open in the light, they open during the dark after the sunset, so that the water can be conserved and the gaseous exchange, that is carbon dioxide and CO2 exchange, it takes place in the night when the stomata open. The carbon dioxide that enters the stomata is fixed in the night and it is used in the photosynthesis during the daytime when the stomata close. So photosynthesis takes place during the daytime, but gaseous exchange takes place during the night, and the daytime opening of stomata is escaped by this procedure. Therefore, scotoactive opening is actually an adaptation in the succulent plants. So, how does the scotoactive opening of stomata occurs? During the dark, stomata open and gases like oxygen and carbon dioxide, they enter the stomata. When stomata close, respiration takes place and starch is converted to pyruvate. This pyruvate forms malic acid with carbon dioxide. With the formation of malic acid, it is transferred to the vacuoles of the cell, of the guard cells, and as malic acid is transferred, the pH of the vacuoles decreases. All the night, the formation of malic acid takes place and thus carbon dioxide is fixed in the form of malic acid and transferred to the vacuoles, and thus the pH of the cytoplasm outside the vacuole, it remains constant, but the pH of the cell decreases. During the daytime, stomata close as light intensity is very high and exchange of carbon dioxide and oxygen stops. But photosynthesis starts in the presence of sunlight, and the supply of CO2 for the photosynthesis is done by the malate that was stored in the vacuoles at night. So what happens? Malate that was stored in the vacuoles is transferred to the cytoplasm outside the vacuoles, where it releases carbon dioxide and forms pyruvate. This carbon dioxide enters the Calvin cycle of the photosynthesis and forms starch. Pyruvate is also converted to starch, and thus in the daytime, when the stomata are closed, photosynthesis takes place with the help of carbon dioxide that is released by the malate. So, in photoactive plants, carbon dioxide is fixed during the night in the form of malate and is released from this malate during the daytime for photosynthesis. Thus, photoactive opening is an adaptation to conserve the water in succulent plants. So, today we studied how opening and closing of stomata takes place in photoactive and scotoactive plants.